So I have now 8 minutes to tell you two rules of brain function, two main rules of brain functions, and show you how we are trying to use this knowledge to modulate the altered brain activity in different severe psychiatric disorders such as schizophrenia or obsessive compulsive disorder. So the first rule that I want to present you is the rule of segregations. So broadly speaking, in our brain, we have cells, group of cells, that are anatomically distinct and seems to support specific functions. The rule of segregation is not new. It was already suggested in 1888 by this man, Ramon Cajal, who discovered that in the brain we have neural elements that will be later called neurons that were anatomically distinct, and what he thought is that they also act independently. Contemporary to this view, there was an opposing view from this man, Camillo Golgi, that he suggested that the brain was actually a continuous reticulum where cells were not acting independently, but they were actually acting together to support brain functions. So we now know that the brain is not a continuous reticulum, but we know that the cells have to act together, they have to share information to actually support brain functions. So today we can study in the living human brain the principle of segregation and integration using this kind of neuroimaging technique. Here depicted is a functional magnetic resonance imaging MRI that we have here at UQ, at the Center for Advanced Imaging. And what this technique allows us to do is to measure brain activity while participants are in the MRI and are performing a specific task or simply resting in the scanner. So thanks to this technique, we now know that there is a specific brain area that actually support specific brain functions. For example, the red region here is a region that is commonly activated or seems to support motor functions. On the other hand, the green regions is a region that is commonly activated or seems to support visual processing. And the blue region here at the top of and the front of your brain is actually a region that is generally involved in a cognitive process such as logical thinking. In addition to study this kind of segregated process, we can also try to investigate how these regions communicate. And a simple way to do so is looking at the neural activity in those regions across time. And the idea is that if regions show a similar pattern of neural activity, these regions are thought to communicate. They are integrated, they share information. In contrast, if the regions are not showing the same pattern of activity, like the red region and the green regions, these are thought to not communicate. We now know, thanks to this neuroimaging work, that psychiatric disorder or brain disorder in general are characterized by alteration in segregated neural activity. For instance, we can have a reduction in neural activity here depicted in this slide from a red to orange color. We can also have the emergence of interaction between regions that should not be there or is not there in a healthy condition, is between the orange region and the blue and the green regions. And we can also have a reduction in how regions communicate or are functionally integrated, and this is what is depicted between the orange and the blue regions. Now, this is very good. We now know what are the neural networks that actually are alterated in psychiatric disorder, but what do we do about that? So there is different options that we have. One option is medication, but as you know, medication carries some severe side effect. Not often, but it could happen. And there is also psychological intervention, but psychological intervention is generally slow, and they require insight from patients that is not often easy to obtain from very sick people. So a technique that we are trying to explore, and other people in other laboratories in Australia around the world are trying to explore, is the use of what we call non-invasive brain stimulations to change or modulate the activity of a specific cortical regions, and by doing so, also modulating the activity of more widespread neural networks. So this technique has already been shown some success to reduce symptoms, but to really, what we really need to do is actually understand how a local change can actually selectively modulate the activity of these large-scale brain networks. And this is an example for a study that we recently performed where we inhibit the activity of motor cortex in healthy subjects. And what we observe is that sensory motor regions that support sensory motor function get more interconnected following these local inhibitions. And these are depicted in purple on the top part of the slide. But they also lose connectivity or integration with the regions that don't support motor functions. In contrast, when we excite the same region of the motor cortex, we don't see this global reconfiguration in brain dynamics. So again, this to stress that we really need to understand how a local change could affect this global brain dynamic to really improve the efficiency of this non-invasive stimulation to treat psychiatric disorder. So in sum, while we cannot change the rule of brain functions, we can actually use them to reshape how the brain functions. So again, the idea here is try to have a specific understanding of what is a network of regions that is actually altered in a specific disorder, and then intervene by modulating the activity of one region to restore the activity of that specific network and eventually restore symptoms of severe mental illness. Thank you very much.